Hello everyone, and welcome to today's webinar in the CERDP and ESTCP webinar series. My name is Rula Deeb. I'm a Senior Principal at Geosyntac Consultants in Oakland, California, and the coordinator of the webinar series on behalf of CERDP and ESTCP. I will be facilitating today's call. The webinar will consist of a brief overview of CERDP and ESTCP, followed by the technical portion of the event, which focuses on the management of 1,4-dioxane contaminated sites. First, Dr. Pedro Alvarez, a professor at Rice University, will describe the development of catabolic gene probes used to quantify the presence and expression of 1,4-dioxane biodegradation rates. Pedro's presentation will be followed by a brief Q&A session. He will be followed by Dr. David Adamson of GSI Environmental, who will present field and laboratory research to inform the conceptual site model of 1,4-dioxane behavior and implications for the management of 1,4-dioxane contaminated sites. Dave's presentation will also be followed by a Q&A session, and we will conclude the webinar with an interactive Q&A session with both of today's speakers. Today's broadcast will be listen only. You may submit questions by using the chat box on the left-hand portion of your screen. You do not need to wait until the Q&A period to submit your questions. In fact, we do encourage you to submit questions well in advance of the Q&A session. On this slide, we have provided a few suggestions in case you experience difficulties with the broadcast audio. Typically, any delay will be fixed if you refresh your screen or call into the conference line shown here. However, if you continue to have problems, please submit a comment using the chat box in the lower left corner of your screen. With that, I would like to introduce Dr. Andrea Leeson, who is the Deputy Director of CERDP in ESTCP, as well as the Program Manager for Environmental Restoration. Andrea has been with CERDP and ESTCP since 2001, and before that, she was a research scientist at Battelle Memorial Institute, where she conducted research on in situ bioremediation. Andrea?
Thanks, Rula, and welcome everyone to CERDP and ESTCP's webinar today. Before we jump into the webinar, let me give you just a little bit of an overview of the CERDP and ESTCP program. CERDP is our Strategic Environmental Research and Development Program, and it was established in 1991 by Congress and is meant to be a partnership between the DoD, the Department of Energy, and the EPA. And our mission is to identify and address high-priority environmental science and technology opportunities that focus on the DoD requirements. And we fund both fundamental research as well as advanced technology development, with the end goal being to impact real-world environmental management. ESTCP is the Environmental Security Technology Certification Program, and under this program we demonstrate innovative environmental and energy technologies. And these investments capitalize on past investments under CERDP or other research programs, and they're designed specifically to transfer the technologies out of the lab and into the field. Especially important for us in our ESTCP demonstrations is to ultimately transition to implementation and regulatory acceptance. Now, CERDP and ESTCP are complementary programs, with much of the CERDP research occurring at the lab and pilot scale, although we do occasionally do field efforts there. And then ESTCP demonstrations are primarily at the pilot and field scale, although we do occasionally have some supporting lab efforts under that program as well. We have 4 program areas in CERDP, 5 in ESTCP. The Energy and Water program area is only in ESTCP, while our other 4, which is Environmental Restoration, Installation, Energy and Water, Munitions Response, and Resource Conservation and Resiliency, those are under both programs, and they're managed jointly by a specific program manager. Our webinar today is focusing on research and demonstrations that were conducted under the Environmental Restoration Program area. Environmental restoration has essentially 5 main areas of research. We look at contaminated groundwater, contaminants on DoD testing and training ranges, contaminated sediments, wastewater treatment, and risk assessment. Our webinar series is highlighting research and demonstration efforts from each of the 5 different program areas. And as you can see here, this is a list of upcoming webinars over the next few months. We cover a really wide variety of topics. Our next topic under environmental restoration will be on August 9th, and we're focused there on some of the new work we've been looking at on wastewater treatment systems for forward operating bases, as well as for fixed installations. Registration is open now for all of these webinars through the end of the year, so there's even more here that are not able to be listed on that slide. You can go to this website here and register for the other webinars. I'd also like to just briefly mention, I'm very pleased to say that we also will have our CERTIP and ESTCP Symposium again this year, November 27th through 29th. It will be at the same location as we had the previous year at the Washington Hilton Hotel in Washington, D.C. in the DuPont Circle area. You can register now for the symposium, and if you go to our website, certip-estcp.org, you'll find a link there that will take you to the symposium page where you can register and get some information about the tech sessions we'll have available. So at this time, Rula, I'll turn it back to you, and thank you.
Thank you, Andrea. It is now my pleasure to introduce Dr. Pedro Alvarez, a professor of civil and environmental engineering at Rice University and the director of the NSF Engineering Research Center on Nanotechnology-Enabled Water Treatment. Pedro's research interests include environmental implications and applications of nanotechnology, bioremediation, fate and transport of toxic chemicals, water footprint of biofuels, water treatment and reuse, and antibiotic resistance control. He received a Bachelor of Engineering degree in Civil Engineering from McGill and master's and doctoral degrees in Environmental Engineering from the University of Michigan. And Pedro was recently elected to the National Academy of Engineering for his contributions to the practice of bioremediation and environmental nanotechnology. Pedro?
Thank you, Rula, for that kind introduction. I want to thank you, Andrea, and CERTIP for this opportunity to present some research that could lead to important improvements associated with the management of large and dilute dioxane plumes. Let me begin with some background. As you all know, 1,4-dioxane, or dioxane for short, was commonly used as a stabilizer in some chlorinated solvents, such as 1,1-TCA. And dioxane has become sort of like the MTBE of the chlorinated solvent plumes, in the sense that it is a relatively recalcitrant molecule that is hydrophilic and highly mobile in groundwater. And it forms large and dilute plumes that are a significant challenge to clean up. And dioxane contamination is pretty widespread. This research here that was led by Dave Adamson, our next speaker, found that dioxane was detected in samples from about 21% of close to 5,000 public water systems at levels higher than the minimum reporting limit of 70 parts per trillion. And close to 77% of the systems had dioxane above the reference concentration, which is 0.35 micrograms per liter. This project is about enhancing our ability to quickly assess the suitability of monitored natural attenuation as a remedial approach. And as you know, natural attenuation refers to various processes that reduce contaminant concentrations and attenuate their migration, including volatilization, sorption, dilution, and mainly biodegradation. And the combined effect of these mechanisms that act without human intervention is known as natural attenuation. Of course, natural attenuation is the most cost-effective approach to manage large and dilute plumes. But the burden of proof that it is an appropriate solution lies on the proponent. And we often document that natural attenuation is proceeding satisfactorily by quantifying the loss of pollutants at the field scale, and of course the associated geochemical footprint. However, this can be very costly and time-consuming because it requires obtaining and analyzing data, lots of data, over large spatial and temporal scales. One emerging and simple approach to support these efforts is the use of genetic biomarkers. So regarding bacteria, for example, it can help us determine who is there, what can they do, what can they degrade, what are they doing, and who is doing what. So along these lines, the objectives of our research is to develop and validate gene probes to quantify dioxane degradation capacity in order to support decisions to select or reject natural attenuation and assess bioremediation performance. And we have 4 objectives. The first one is to assess the reliability of a biomarker that we developed for a bacterium, Pseudonocardia deoxyoboran CB1190. This is the archetype for dioxane degradation. And we also are discerning site-specific geochemical or contamination scenarios that influence the relative abundance of these specific degraders and their activity, as well as to isolate or identify novel dioxane degraders and characterize novel catabolic genes so that we can enhance our forensic analysis capabilities of degradation potential. Now, when we started our research, the genes responsible for dioxane degradation were unknown. But previous researchers like Shaili Mahendra working with Lisa Alvarez-Cohen have discerned that the attack was initiated by monooxygenase enzymes, as shown to the left of this slide. And we already have the sequence available for CB1190. So using bioinformatics and transcriptomic analysis, Meng-Yang Li identified a soluble diiron monooxygenase operon that is responsible for initiating dioxane degradation by CB1190, which we call dxmA, or some people call it thmA because this gene also calls for tetrahydrofuran degradation. And the point here on the bar graph on the right is that this gene was induced by both dioxane and tetrahydrofuran, which we confirm by reverse transcriptase quantitative PCR. And so we proceeded to develop a primer probe set to target this specific dioxane monooxygenase gene and measure its concentration using TACMAT chemistry, which gives us more reliable results. And after doing some sequence alignment, we developed the probe, which is written in red letters. That's the probe that we use to target this gene sequence. So regarding our first objective, the first thing we did is we went out there and got groundwater samples from multiple contaminated sites in different states, and we measured the dioxane degradation activity in microcosms. And we also measured the concentration of this catabolic gene, and found a positive significant correlation between gene copy numbers and degradation rates, suggesting that we might be able to predict biodegradation rates within an order of magnitude based on this type of molecular analysis. Now, in contrast, to the right, you see another line where there's no correlation. What I'm talking about here is that if you use total bacteria concentrations assessed by a generic biomarker for the 16S ribosomal RNA genes, you do not get a correlation to degradation activity, which corroborates the selectivity of our thmA dxmA probe. And recently we analyzed additional samples from a site in Washington that had no degradation activity, and we did not detect the dxmA biomarker. So the good news is that so far, this biomarker has never yielded a false positive. Now, what that means in practical sense is that if you find this biomarker present, there is significant potential for dioxane degradation. But if it is not present, that does not rule out that there could be other bacteria with other genes that could degrade dioxane. Regarding our second task, we conducted a multivariate correlation analysis to discern general site conditions that are more amenable or least amenable to natural attenuation. And this analysis showed that if oxygen is not limiting, higher dioxane concentrations are more amenable to biodegradation, presumably because they enhance the sustenance of specific degraders and induce specific enzymes. So the practical implication here is that enhanced aerobic bioremediation strategies will be more efficiently employed in source areas where you have higher dioxane concentrations and acclimated microbial communities. In our third task, we're addressing the concern that we often study and work with cultivable bacteria like CD1190, whereas 99% of the bacteria in the environment do not grow on plates, and many of them may be important to natural attenuation. So we need to learn more about them. And we're using, in this case, fluorescent activated cell sorting, which is the bottom right icon. And here we're using a DNA probe to optically label cells that have the catabolic gene of interest, and then we separate them using flow cytometry. And then we have set aside separated bacteria that have the gene of interest. And to learn how to use this method and illustrate it, we first developed a phylogenetic probe for Pseudonocardia species. We call this probe PSE631. And the reason why we targeted these Pseudonocardia is because 18 out of the 34 known bacteria that degrade dioxane belong to this genus, as illustrated on the phylogenetic tree on the left. And we targeted initially a phylogenetic probe because it is easier than targeting a catabolic probe, since phylogenetic genes, are much more abundant than catabolic genes. Typically, there is more copy numbers of them within a cell. And so we use in this method development exercise CD1190 as a positive control because this is a Pseudonocardia. And we use several negative controls like E. coli, Bacillus subtilis, and even some dioxane degraders like mycobacteria that are not Pseudonocardia. And we got pretty good results. You can see on the left the so-called scattergrams. And in green, we see successful separation of the positive control, but not the negative controls like E. coli. And then we verified that we were separating the right bacteria using quantitative PCR. But the main point here is that this culture-independent cell separation approach facilitates subsequent molecular analysis to advance our understanding of potential dioxane degraders. So once we had the method, we switched the probe to the thmA probe in order to separate dioxane degraders that have this gene and try to learn how diverse these are. Are they only Pseudonocardia, or can this gene be found in other species, for example, to understand the propensity for horizontal gene transfer? But there is a challenge. There is only one copy number usually of this gene present in this genome, so that represents a detection challenge. So you need to obtain a sufficient signal to be detected, and we apply several strategies shown in the bottom here. The first one is that we synthesized longer probes to increase the binding capacity. Second, we use 5 different probes simultaneously to target different regions of the gene. And last, we use multiple fluorophores on each probe to enhance detection. And altogether, this worked. And we then took samples from dioxane-degraded microcosms. These ones were enriched from activated sludge from a wastewater treatment plant. And we sorted the cells accordingly with the fluorescent activated cell cytometry and did Illumina sequencing to find out the microbial community structure. And on the left pie, we see that Proteobacterium and Actinobacteria were dominant at the phylum level. On the right-hand side is where the most interesting stuff is. We found several known dioxane degraders, which are the red text. This included, for example, Amphibia and Pseudonocardia. But we also found several other bacteria in blood tests that were not previously reported to be dioxane degraders at the genus level. And 5 of these dominant genera in green boxes belong to the Rhizobiales group. that suggests that the rhizosphere of plants might be a certain environment to isolate dioxane degraders. So we are collaborating with Jerry Schnoor at the University of Iowa to try to isolate some of these degrading bacteria from the rhizosphere. Now, in task 4, we are sequencing the DNA of putative novel dioxane degraders to discern the responsible genes and use this information to either optimize our current probe or design new probes for new genes. And along the way, learn about the diversity and evolutionary relationships of dioxane degrading genes. In this particular example, it is important to evaluate the presence of dioxane degraders in uncontaminated environments to assess the potential for natural attenuation when the spill occurs in a pristine site, for example. And as you can see here, we took samples from garden soils with no known previous exposure to dioxane. This is important. And we were able to enrich them with dioxane and get degradation activity going after a couple of months. And interestingly, as you can see in the right pie charts, mycobacteria were the dominant genus in both consortia after enrichment on dioxane. I should point out that in this case, the thmA gene from CB1190 was absent, which means that there must be other genes that are also important for dioxane degradation. So we were not able to isolate these pure mycobacteria strains from the enrichments. They were a bit fastidious. But we had a pure culture, Mycobacterium pH06, which is known to degrade dioxane and didn't have the thmA gene either. So we proceeded to fully sequence the genome of that bacterium. We named this strain Mycobacterium dioxanotrophicus pH06 and proceeded to identify the gene. By the way, this pH06 offers some potential advantages for bioaugmentation over CB1190. One is, as you can show in the data on the bottom right, is that it has a higher affinity for dioxane, a higher cell yield, and also is subjected to less clumping, as shown in the picture there, which makes it easier to perfuse when you add it to aquifer. That's the problem with CB1190, that it gets filtered out very easily because of clumping. So the first question we made ourselves is whether monooxygenases were involved in the initial attack, as is the case for CB1190. And we use acetylene, which is a known inhibitor of monooxygenases. And as you can see here, when acetylene is added, there is no degradation, suggesting the involvement of a monooxygenase. So we sort of knew what we were looking for. And then, with this insight, we had sequenced already the full genome of pH06 using Illumina and by technologies, and we got lucky. What I mean by that is that the sequence identified only one soluble diiromonooxygenase gene cluster. This happens to be located in a linear plasmid, and it has 4 subunits, A, B, C, and D. And this gene looks a lot like propane monooxygenase genes. There's about 88% similarity. So we named it PRM-ABC, PRM for propane monooxygenase. And we then did some gene expression transcriptomic analysis to make sure that this was indeed induced by dioxane or tetrahydrofuran. And indeed it was. This data shows you induction or upregulation of this gene by dioxane relative to glucose and acetate as negative control. So we knew that we had the right gene, and we proceeded to develop a biomarker for it. And similar to what we did before with CB1190, we essentially proceeded to use alignments of the alpha hydroxylase subunits and for different groups of SDIMOs. As you know, this is the unit that usually is responsible for specificity of these enzymes. And we use the same two criteria. Number one is avoid the diiron centers, which is the conserved active region, which is similar for all monooxygenases. And two, we targeted the surrounding hydrophobic residues, which are the sites that are responsible for substrate recognition and binding. And we ended up with a biomarker that was selective. Again, there were no false positives with bacteria that do not have this gene. And also, dioxane degradation rates by mycobacterium enrichments were significantly correlated to the concentrations of this biomarker. But as was the case before, they were not correlated to 16S RNA concentrations. Now, on a separate note, we discovered something very important here. When both dioxane degrading genes that we know now about, the thmA gene and thmA gene, are on plasmids. And these plasmids can be lost when they are not needed because, bacteria don't carry excess luggage. It, requires a lot of energy to maintain and reproduce plasmids that are not needed. So in this example, when you use auxiliary substrates that could be used to promote the growth of the bacteria when dioxane concentrations are low, you could lose the plasmids. They could get cured, and therefore you could lose dioxane degradation capabilities. So it is very important to monitor the state of the plasmids and quantify the expression of the dioxane degradation gene under different substrate conditions. For example, what happens if you have chlorinated solvents present also, and you want to biostimulate co-metabolism with propane? Are you going to keep or lose that gene and therefore the dioxane degradation capability? So to address such questions, we're planning to use transposon mutagenesis to insert a reporter fluorescent protein just downstream of the promoter of the dioxane degrading gene cluster. And when the gene cluster is induced and the plasmid encoded protein regulator is made, then the reporter gene will be expressed as well, and the fluorescent signal can be used to monitor the dioxane degradation capabilities and choose appropriate bacterial enrichment and biostimulation strategies, and of course avoid conditions that lead to plasmid loss. But anyways, maybe this is a good time to begin wrapping up. To summarize, we have shown that our biomarker for dxmA conservatively indicates dioxane degradation capability. We don't have a single false positive yet, but false positives are possible, meaning that there could be bacteria that are present that degrade dioxane that cannot be detected. We see that dioxane degradation is much more likely when it's present at higher concentrations that sustain and induce dioxane degraders, provided that oxygen is available. This is very important for fast degradation. We also saw that a fluorescent activated cell sorting can isolate and separate genomes of unknown potential degraders so that we can learn more about them. And that dioxane degradation can occur even in uncontaminated soil, so it might be more widespread than previously appreciated. We saw that mycobacterium species dominated the enriched consortia from this uncontaminated soil and discovered a new catabolic gene in a mycobacterium species, PH06, and successfully designed a probe for it. Very quickly, I believe that many dioxane plumes could be effectively managed by monitored natural attenuation at a small fraction of alternative treatment costs, provided that we can demonstrate that natural attenuation is an appropriate solution for that particular site. So this project is contributing a simple forensic tool to enable these decisions to either select or reject thmA, and also to assess the performance of bioremediation. Very quickly, this is the team that helped me obtain the data that I presented to you today. And I want to leave you with this slide where you can find additional information and details on this presentation. So thank you for your attention, and Rula, back to you.
Thank you, Pedro. I would like to remind all of you to submit questions by using the chat box in the left-hand portion of the screen. We have received a number of questions for you, starting with the following. Can you please elaborate on why 1,4-dioxane is recalcitrant in subsurface environments, Pedro?
Yeah, I think this is mainly due to its chemical structure. Usually, bacteria degrade easily compounds that look like their natural diet when they have a common building block. Dioxane, as you know, is a diether. It has two ether bonds. And ether bonds are rare building blocks. They are rare in the things that bacteria normally use. But if challenged, when bacteria have nothing else to eat, obviously they evolve capabilities to degrade dioxane. I remember when I was a student, I was told that this was one compound that would never be degraded, as was MTBE. And by now, we keep finding dioxane degraders even in clean soil, as we do here. So it is not clear if it is that the bacteria are evolving and transferring these genes horizontally and spreading these degradation capacities, or that we're getting better at finding them. It could be a little bit of both, but it is indeed a recalcitrant compound, mainly because of its chemical structure.
Thank you. So based on the results of your work, can you tell us whether the presence of catabolic genes guarantees that dioxane will be biodegraded?
That's a good question. The truth is no, because the presence of a gene does not necessarily guarantee its expression. It doesn't mean that the enzyme will get made and the enzyme will be active. But however, if at a site you see elevated concentrations of these genes compared to the background, that tells you that bacteria that can degrade dioxane are growing. And that is a strong circumstantial line of evidence that if there is growth, there is also degradation. Of course, you need to consider other evidence. For example, is there concentration loss? Is the geochemical footprint consistent with biodegradation? But overall, over the long spatial and temporal scales that are relevant to natural attenuation, the enrichment of catabolic genes associated with a decrease in the concentration of the contaminant should be evidence of biodegradation.
Great, thank you. Does your work mean that we need to be careful about biostimulation approaches involving primary or auxiliary substrates to enhance the growth of 1,4-dioxane degraders?
Yes, I think so. And it's a difficult balance here because sometimes in biostimulation, of course, you need to make sure that the bacteria that you want to live are thriving, and you don't want to give a competitive advantage to other bacteria. But the problem here is that if you are giving bacteria something easy to eat and that they can metabolize through their central metabolic pathways, then they don't need those genes that are coded in plasmids. And since it takes a lot of energy and resources to maintain those plasmids, when bacteria grow and replicate, they leave them behind and they lose the plasmid. So what we were seeing is that, for example, using butanol, which could induce some monooxygenases that could co-metabolize dioxane or chlorinated solvent. When bacteria grew on these compounds, they lost the plasmid in the absence of dioxane. So yeah, I think it's something that has been sort of overlooked so far, and needs to be recognized that when catabolic genes are on plasmids, they could be lost if there is no competitive advantage to keep them.
Great, thank you. Can you please tell us if there are plans to commercialize the biomarkers you developed, and if there are other similar products that are available for use by our audience?
Yes. Well, we have a patent pending. The preliminary patent has been approved. And I believe that in the future, this sort of analysis, molecular-based analysis, are going to become more common because, for one thing, synthetic biology is very rapidly evolving, and the costs of these analyses are relatively low. You can, with a few $100s only, find out a lot, even have inference on the relative amount of activity that you can get. So I do foresee, a significant increase in this sort of activities. I personally am not interested in business. I'm more interested in the science, but I would be happy to help anybody that is interested in this sort of activities.
Great, thank you. One more question for you. What options are there to remediate a 1,4-dioxane contaminated sites if the biomarkers are not found for specific degraders at that site?
Well, so let me emphasize that if the biomarkers are not there, that does not mean that there are no potential degraders. That just means that those specific degraders are not present. There could be others that we don't know about. But I think that if we're talking about a source zone, there are several options. One could be bioaugmentation. That means adding bacteria that can basically fill the metabolic niche that has not been filled. And of course, we need to be careful that we consider bacteria that are safe and that are easily dispersed and have good perfusion characteristics. But of course, other things that could be considered include maybe co-metabolism or even in situ chemical oxidation if site-specific conditions are such that the contamination is well constrained and easily accessible, and we know where it is. That's part of the challenge. A lot of times we don't understand that well due to heterogeneity where the source zone is.
Great, thank you. Back to the use of the biomarkers. Do you envision them being used as a one-time tool when you're making decisions about using M&A, or would they be used as part of a routine monitoring program?
Yeah, I would because of the low cost of the analysis, I would do it frequently to, for example, make sure that the populations of these dioxane degraders are being sustained, that they're not disappearing or dying off. In fact, we want to see that they're increasing with time near source zones. So I would do it as part of the initial characterization and maybe perhaps annually or even quarterly, depending on what the regulatory agencies want in terms of monitoring. But it's relatively a simple add-on that you would do as you also measure other things such as dioxane concentrations.
Thank you, Pedro. Another question from our audience. You suggested that propane may inhibit the plasmid-coated degradation capacity. Is this currently being tested further? And if so, will the results be available soon?
Yeah, no, I apologize. I didn't say that it wouldn't inhibit. I said that it is important that we consider, that we look at it, whether propane would inhibit or not. We do know that propane can induce propane monooxygenase, and many times this is beneficial in terms of co-metabolizing dioxane. But we need to be careful that bacteria just load propane and not losing the plasmid for dioxane. So this is something that we are looking into, and we hope to be able to report that within 6 months.
One last quick question before we move to Dr. Adamson's presentation. Have you done any work to look at pH and dissolved oxygen effects? Is there an optimal pH range or dissolved oxygen level for 1,4-D biodegradation?
Yeah, in my opinion, aerobic conditions certainly facilitate biodegradation. So here, of course, going, above 1 milligram per liter would be nice. There is some evidence, somewhat circumstantial, that biodegradation can occur under anaerobic conditions. It's certainly thermodynamically feasible, but we have never seen reproducible, solid evidence that this is a sustainable and sufficiently fast process yet. But I think that the jury is still out on that. As far as pH, that would be species specific, but most bacteria like to be between pH 6 and 9, but I wouldn't rule out that there is acidophiles, for example, that prefer to degrade. I know that for BTEX, some bacteria love eating BTEX at pH 2 in landfills, but that's rare. I mean, the norm probably would be stay near neutral would be better.
Great. Thank you so much, Pedro. We'll get back to you in the final Q&A. But at this point, we'd like to go ahead and transition to our next speaker, who is Dr. Dave Adamson. Dave is a principal engineer with GSI Environmental in Houston, Texas. And since joining GSI in 2004, he has focused his practice areas on research and development, site investigation, characterization, and remediation. He has extensive expertise in natural attenuation, source zone characterization, matrix diffusion, and the testing of innovative treatment technologies. Dave has served as a PI on multiple DoD-sponsored research projects. He earned his doctoral degree in civil and environmental engineering from the University of Iowa and is a licensed professional engineer in the state of Texas. Dave?
All right, thanks, Rula. Well, today I'm going to present results from a CERTA project that we completed. It's CERTA project ER 2307. One of the goals, the primary goals of this project, we want to take a closer look at some of our assumptions about what 1,4-dioxane sites look like and some of our assumptions about how it really behaves in the environment. And what we found out, the results surprised us a little. In part, we found out that some of the assumptions didn't end up to be necessarily all that well-founded, and meaning that conventional conceptual site model for 1,4-dioxane needed some adjustment. So that's what I'm going to focus on today. Before that, let me acknowledge that I had a lot of collaborators on this project, including Shaylee Mahendra and her group at UCLA, Mike Wong and his group at Rice University, Dan Bryant, Woodard Curran, and Chuck Newell and a whole host of others here at GSI Environmental. And those guys did a lot of great work, a lot of which I can't cover in this short talk, but it's making its way into other articles that will be published soon. So in terms of what I'll talk about today, we've got this agenda. I'm going to go through a bunch of data related to 1,4-dioxane and trying to build that proper conceptual site model. So you see some of the ways that those are addressed here. At the end, I'm going to come back and discuss some of the implications of these results for site management strategies. So I mentioned that there's sort of a traditional CSM for 1,4-D and CVOC sites. So what does that look like? The graphic here tries to lay some of that out. We're looking at a plan view of plumes that might result from a co-release of 1,4-dioxane and chlorinated solvents, something like 1,1,1-TCA or its byproducts. So you've got an easily identifiable source zone over there on the left, and then the chlorinated solvent plume, that light red part, extending down gradient, and then eventually sort of peters out. The 1,4-dioxane plume, on the other hand, in this theoretical case, extends farther. 1,4-dioxane, it's really soluble, doesn't strongly sorb, and it's always, as we've sort of talked about, been thought to be relatively recalcitrant, particularly relative to chlorinated solvents, and then also not necessarily all that impacted by any historical remediation efforts that might have gone on the site, like enhanced bio or something that would have probably created unfavorable redox conditions anyway for its degradation. So collectively, all these properties, they sort of lead themselves to these assumptions that 1,4-dioxane plumes will be long relative to the chlorinated solvent plumes. And if this CSM is true, it's not really a pretty picture. The problem with it, though, is that all of this is sort of based on perception. Nobody's really gone out there, collected data, and tried to actually validate, technically validate whether this is true or not, and so that's what we attempted to do as part of this study. So our overall objective, improve the technical basis for conceptual site model development, hopefully use that sort of to inform better management strategies for sites with 1,4-dioxane and CVOCs. We went about this in a bunch of different ways, including big data studies, proof of concept modeling, a little bit of our own field data, as well as a lot of bench scale treatability testing. I think the big data studies in particular, they provided some key insights that I'm going to focus on today. We're looking at data from lots and lots of DoD, commercial industrial sites, and we're trying to identify sort of broader patterns in these, rather than just sort of basing our expectations on our own experience that maybe only at a few different sites, and at sites that may or may not really be representative of what's really out there. So what we tried to do in terms of our approach for this is basically, sit around and list, what do we think some of the commonly held hypotheses about how 1,4-dioxane would behave in the environment, and then figure out how ways we could test those hypotheses. So that's what's shown in this list right here, sort of a partial list of what we came up with, and it includes the ones that I'm going to talk about today. I'll go through these one by one through some slides, and by the end we'll be able to fill in this column on the right, whether or not the data support the hypotheses or whether we need to rethink our conceptual site model. So to start off with, trying to first take a look at what we saw in terms of looking at these data of what a sort of typical dioxane plume looks like. So we used a lot of geospatial analysis in this. We took groundwater monitoring data mostly from the California GeoTracker database. And when we did these analysis, we came up with a medium plume length for 1,4-dioxane plumes of around 880 feet. And that's based on data from 109 sites. The complete distribution of plume lengths is shown in that box and whisker diagram on the right. And there are some long ones, but again, the median was not even a quarter of a mile long. What's also striking was that you look at the maximum concentration at these sets of sites, it's generally pretty low. The median of all these site-wide maximums was about 370 micrograms per liter, so pretty low. If you add in the 80 or so sites where we did see 1,4-dioxane, but we couldn't really establish a plume length, then it drops even lower, something in 100-something micrograms per liter. So these results really support a conceptual model that the large majority of plumes that are out there they're relatively dilute, and that you're not necessarily going to expect to find milligram per liter concentrations that you might associated with the source zone. That would be a more rare condition. It's also important to note that 1,4-D, basically we're establishing that it co-occurs with one or more chlorinated solvents at virtually all the sites, 95% of them, and that most sites reported at least one form of remediation. Okay, so how do the plume lengths then shake out in terms of looking at them at sites where you also had chlorinated solvents? So this particular graphic is showing the distribution. It turns out that at sites where both of them were present, 105 sites in this particular case, the 1,4-dioxane plume was estimated to be longer than the chlorinated solvent plumes at only about 21% of the sites. So even though we thought that would be the majority of them, it turns out not to be the case. Really, at 56% of the sites, the chlorinated solvent plume was actually longer, and at 23% of them, roughly the same. So these results are really running counter to that conventional CSM we talked about before. They sort of suggest that the 1,4-dioxane plumes are less likely to extend beyond current monitoring networks than we sort of originally feared. We've had a lot of good discussions about why the data look like this. There's some evidence, that natural attenuation or the implementation of pump and treat systems have an effect. But there's this also important thing to remember about how these solvents were used at these sites, and TCE was definitely used at a lot of these sites, and sort of have a head start in terms of 1,4-dioxane that may at a lot of these be associated with the use of 1,1,1-TCA, which might have happened several decades later. Another important thing is that at most of these sites, the chlorinated solvent that's out there far is something like TCE or 1,1-DCE, and 1,1-DCE itself is relatively mobile as well. So this is sort of what the data looked like when we did the analyses. It's not to say that this is necessarily the way it's going to remain. 1,4-dioxane is obviously mobile, and we'll have a chance to catch up at a lot of these sites. Another question we wanted to ask is, are dioxane sites poorly delineated? There's several different ways to sort of address this type of a question. The data on the next couple slides look at the extent to which we're failing to even identify 1,4-dioxane sites. So we're looking at co-occurrence data for 1,4-dioxane and various chlorinated solvents. So you got TCE there on the left in yellow, that big bubble showing that it's detected at 2,322 sites. 1,4-dioxane was detected at around 180 of these TCE sites. And at 168 sites they analyzed, or 169, excuse me, analyzed for 1,4-D but didn't find it. So that means that 1,4-D is detected at about 52% of the sites that contain TCE. For TCA, here in the middle, orange in the middle, 1,4-D was present in about 70% of the sites, and a similar percentage we saw for 1,1-DCE. And again, we're showing, we don't show the data here, but virtually all the 1,4-D sites had one or more chlorinated solvents there. These data are really similar to some other co-occurrence studies. Hunter Anderson had a great paper on this earlier. What's sort of different about these data is these other bubbles. So we're trying to identify sites where chlorinated solvents have been detected, but 1,4-dioxane hasn't even been analyzed for. So for TCE, if you look at that number, that's 1,973 sites where TCE has been detected, but they haven't analyzed for dioxane. So on a percentage basis for these, this is around 67 to 85% of all these sites for these particular constituents. You look at TCA, no dioxane analyses at 318 of those sites, basically 2/3 of those sites. And so if you look at sort of the co-occurrence patterns and you do the math on that, that sort of works out to around 239 sites where we'd expect to see 1,4-D, but nobody had even attempted to measure for it yet. Take this a step further, you can look at sites where only the byproducts of TCA are present, so 1,1-DCA and 1,1-DCE. TCA degrades pretty rapidly, and you can imagine at these sites there might not be as much of a reason to think about the potential for 1,4-D to be there, even though we do know it co-occurs with these compounds as well. So on this one, if you look on the left one, there's 572 sites with 1,1-DCA, but non-detectable levels of TCA, where they never have analyzed for 1,4-dioxane yet. DCE, 1,1-DCE, the case is 59 additional sites. And so we know that most of these will have 1,4-D, and if you start adding these sites up along with the TCA sites on the previous slide, it's clear that we're talking about hundreds and hundreds of sites. These are California data, hundreds and hundreds of sites alone in California where 1,4-dioxane plumes have yet to be identified and characterized. So then when we start asking ourselves questions about attenuation, whether we still consider it recalcitrant, whether there's evidence for attenuation, and Pedro already presented a pretty good case in his talk, so hopefully I don't have to convince you too much that it really is attenuating, but let's see what the data say. So this chart is showing the amount of concentration reductions of individual compounds over time, and we're expressing it as the reduction, the order of magnitude reduction, basically the ratio of the current concentration to the maximum concentration at the site. So for TCA, we saw about a 0.94 order of magnitude, basically right around 1, about a 90% reduction. That's not too surprising since we know that's pretty degradable. What's interesting is that 1,4-dioxane, we're seeing that at 0.5 ohms, or about 75% reduction. And that's really similar to what we saw for TCE, 1,1-DCE, and 1,1-DCA. In fact, statistically, you can't really distinguish between those different reductions. The only one that's really clearly different, attenuating faster, is TCA. So these data suggest that not only is 1,4-D attenuating, but it's attenuating at a similar degree as most of the other chlorinated solvents that might be present. To look at this closer, we want to sort of look at rates, and so we teamed up with Hunter Anderson at AFCEC, and we looked at this in a couple different ways. First, on the left side, we're estimating source attenuation rates of California geotracker sites and determine which ones of these are statistically significant. So that's what's shown in this histogram with a frequency distribution of the dioxane half-lives on the x-axis. And then Hunter did this analysis of the Air Force data. He's using monitoring wells in this particular case. But in both cases, we're really able to establish that there's significant dioxane attenuation that's occurring, and it's happening at a meaningful number of locations. It's not all sites, in part because we're sort of setting these statistical thresholds, so that knocks out a few sites. But at the ones where we do see it, so on a site-wide basis on the left, we saw a median dioxane half-life of 20 months for those 22 sites. At the wells on the right panel, the median value for those corresponds to a half-life of about 48 months. So that's pretty significant in terms of sort of building a case for attenuation. Hunter also, is a really good statistician, and he wanted to take this an additional step further. He used linear discriminant analysis to try to identify the effect of various factors that might influence 1,4-dioxane attenuation patterns in that Air Force data set. So he looked at a lot of stuff. What he's shown here is the effect related to dissolved oxygen metals and CVOCs. That canonical structure on the y-axis, you've got to think of that as sort of a relative measure of the effect. The bigger it is, the bigger the effect. That effect basically beneficial if it's plotting greater than 0, and inhibitory if it plots less than 0. For these particular sets of data, dioxane attenuation could be positively correlated to the presence of oxygen and negatively correlated then to the high concentrations of chlorinated solvents as well as metals. And these are the sorts of patterns we sort of expected to see. If you look at all the great research that's been done in the lab at a lot of different universities, that's what they would expect to see based on the degradation patterns that we're seeing. So this is a good confirmation that these field data, the attenuation that's happening there, is really due to potentially biodegradation. The next question is to sort of understand if we weren't seeing attenuation at the site and we needed to consider remediation, what treatment trades would be the way to get there. And for example, you're asking yourselves, could you use one technology to address part of the contaminant mix or even improve the efficiency of the overall process? Which things are compatible, which things are not compatible? So this testing was done largely by the research teams at UCLA and Rice. And they were looking at a whole bunch of different combinations, including the ones that are listed here on this slide. Essentially, they're taking material from a field site, AFP3 in Oklahoma. They're doing bench scale microcosms where they're monitoring changes in contaminant concentrations and microbial populations over time. And they swapped their systems back and forth, and it worked out pretty well in terms of their testing. We did a lot of these, as I mentioned. I've got limited time, so I'll focus on this one. This is the oxidation followed by biodegradation. So the chemical oxidation in the first step, we figured this is most worthwhile to sort of examine even before we started, because we know a lot of sites may have already done it, maybe not intentionally, because they're only worried about chlorinated solvents. But secondly, we do know that also offers some big advantages. If you've got ISCO, you can potentially take out the CVOCs, and we know that these are inhibitory to 1,4-dioxane biodegradation. So those organisms then are sort of freed up to work. And so we got a lot of data. This is just an example of sort of some of the stuff that we found. These are 1,4-D concentration versus time data for 5 different treatments. The treatments were variable based on whether there was high or low concentrations of 1,4-D in the CVOCs that were present initially. So that's what the y-axis is showing. That initial step, the ISCO step, we basically used that to remove approximately 50% of the dioxane that was present, and that's shown in that sharp drop before day 0. We used hydrogen peroxide as the oxin in this case, so that's relatively short-lived, gone within about a day. So day 0 starts that biodegradation step. And you can also sort of think of that second step as natural attenuation. But one thing that we did was also bioaugment with CB1190 in one of these cases. And that turned out to be pretty important to include bioaugmentation because as you can see by the relatively straight lines in the other treatments, there really wasn't much 1,4-D degradation occurring by the native culture. CB1190, on the other hand, it was really able to thrive even after exposure to the oxidant. It also worked because the oxidant removed all the chlorinated solvents. You no longer had to really worry about these inhibitors of 1,4-dioxane biodegradation, and the culture was essentially able to degrade multiple spikes of 1,4-dioxane after that. So in the absence of chemical oxidation, we saw sort of the opposite pattern. The undegraded chlorinated solvents completely inhibited 1,4-dioxane biodegradation. Okay, and a final thing that we looked at then that I'll discuss here is trying to understand the potential impact of matrix diffusion at 1,4-dioxane sites. So this is something that we know that's important for chlorinated solvents. We know it basically contributes to long-term persistence of these contaminants because what happens is that they basically over time get stored in these lower K zones. So that sort of top panel of that graphic shows you've got a source present, and as it's present, those concentration gradient that's created sort of pushes material down into, in this case, an underlying low permeability zone, something like a clay or a silt. Once that source is depleted, shown in the bottom panel, you've sort of reversed the concentration gradient such that any mass that's in that low K zone can sort of then diffuse back out and contribute to long-term persistence within an overlying transmissive zone. So for dioxane, if you're thinking about 1,4-matrix diffusion, you might not think it's all that big of a deal because it's a pretty soluble compound. It doesn't necessarily absorb all that long. Maybe the loading period's relatively short. But since it's present at high concentrations, there is a potential that it could exacerbate effects because you have a relatively high concentration gradient to cause diffusion. So we set up a bunch of proof of concept modeling studies. What we used was the matrix diffusion toolkit. This is developed in part by GSI for ESGCP. And we modeled a release of 1,4-dioxane TCA in 1970. We set this up as a pretty simple two-layer system. That's what's shown here. Basically a sand unit, that transmissive area in the top, a low-K layer on the bottom. Then the x-axis then shows moving down gradient into the plume. In these cases, we release a source on the left in the sand, and then groundwater moves from left to right. And the big question then is sort of what happens in that lower unit? What happens in the low-K unit? And so we track that basically over about an 80-year period. And so a couple of results slide. This first one is basically showing snapshots of the mass distribution of 1,4-dioxane, that's on the left, and TCA on the right, within the low-K zones extending down gradient into the plume. So the concentration legend is down there, but basically the yellows, orange, reds colors, those are the high concentrations, and the blues are sort of the low or non-detect areas. What happens on this is that you realize sort of right away that 1,4-dioxane, the source itself, the amount that stays sort of close to that release point, it's actually relatively short-lived. In this case, depleted within about 3 years, and that's why we show the 1973 snapshot there. But the thing is that the concentration is really high initially, a really soluble compound, and that creates a really big concentration gradient. It's pushing a lot of mass down there initially, even though it's relatively short-lived. By 2010 and 2050, most of that mass has moved in the transmissive zone out of the source area. But what it's done is it's allowed a very large portion of the plume, the low-K zone underneath the plume, to also have relatively high concentrations of mass. And so it's penetrated deep and it's penetrated long into the plume, and especially relative to what you're seeing with TCA over there on the right hand, where the concentrations aren't nearly as high and the penetration is not nearly so deep. This second graph is basically showing a concentration trend in a source zone well screened within the transmissive zone. So immediately above the low-K zone within the source area. It's only plotting data after the source is gone, so it's directly representing the contribution of back diffusion. So even 88 years after the release, the 1,4-dioxane concentration resulting from this back diffusion, it's still above 0.1 milligram per liter, somewhere between 0.1 and 1 milligram per liter, and relatively similar to where you see TCA after that point. The problem becomes that TCA is not too much different than sort of maybe where the MCL is, but 1,4-dioxane after 80 years, we're still left with a back diffusion signal that's several orders of magnitude over the sort of ppb levels that you potentially are seeing in terms of applicable regulatory criteria. So these data are clearly representing that 1,4-dioxane is subject to matrix diffusion, and it can really contribute to long-term persistence. So in terms of filling out our original hypotheses, we found that dioxane plumes are relatively dilute, but not necessarily large. In terms of when we're looking specifically at co-occurring plumes, the evidence pointed that they're not necessarily larger than the chlorinated solvents plumes that they're co-occurring with. Dioxane plumes are poorly delineated? No, we actually didn't present that data. Most of them were relatively well delineated. What we saw is a bunch of potentially unidentified plumes. We saw lots of evidence for dioxane attenuation, in many cases similar to the chlorinated solvents. Moving on to the next set, geochemical conditions and co-occurring contaminants influence dioxane attenuation. We did see some patterns. None of them were necessarily all that predictive, but we did see patterns that sort of matched up the field data with what we'd expect, sort of based on the lab data. Treatment trains, they were definitely demonstrated as being effective, particularly critical in terms of removing co-occurring contaminants. And matrix diffusion, the assumption is that it wouldn't necessarily be an important process because of the solubility of dioxane. The modeling data, and we did collect some limited field data, confirmed that it would contribute to long-term persistence. So the data collectively that we collected, one of the primary benefits that we see out of it is it's really building a much stronger case for M&A relative to when we started. And Pedro also presented some great data that shows that as well. We've got generally dilute and plumes that are often similar in size to co-occurring chlorinated solvents. We've got a lot of evidence for attenuation. And as Pedro mentioned, we've got lots of new ways to basically collect the lines of evidence that we need to demonstrate M&A. In terms of conclusions, there are some overarching things that we found out, one of which it's sort of become apparent to us is that dioxane source zones, they're not really like other source zones. In most cases, these are dilute sites. Dioxane's been depleted out of the source zone, and you may not really have an easily identifiable target in order to sort of treat with traditional technologies. The persistence that you see, sort of these dilute plumes, or potentially dominated by secondary sources, things that are resulting from matrix diffusion processes. And these sort of match up with that idea of what we see with our modeling and also with the data studies that show that these are mostly dilute sites. So this process, this matrix diffusion process, is something that you really need to include in terms of developing a site-specific conceptual site model. It also falls then from these, if this is what a typical site would look like, traditional source treatment is not going to necessarily be an easy thing to do, not necessarily very effective in terms of treating up mass and low K zones, and not necessarily very cost efficient if it's a dilute plume. So if that's the case, site management in a lot of instances might need to focus on longer term management, maybe targeted treatment, but really trying to look hard at whether you can build a case for M&A being a viable option at your site. If you do find yourself in a place where treatment is required and you can find something that's sort of a source zone that's easily identifiable, something you can target, then treatment trains, as these data show, are a potentially good option, particularly because of their ability to remove co-occurring CVOCs. So that's what I have. My information is here below, and the project page you can find a lot of good information. The final report for this project has been released and can be found there. And there's a number of publications as well. So thanks.
Great. Thank you so much, Dave. We have a number of questions for you. You mentioned that you collected field data, but that you didn't show any of these results today. What relevant findings can you share with us?
Yeah, so we went out, and I guess I should clarify that we did collect some of our own field data as part of this project. We went to Cape Canaveral Air Force Station, and what we're trying to do there is basically take a look at whether there was evidence for matrix diffusion at this particular site. And that's a site where a lot of different work has been done in terms of trying to remediate chlorinated solvents, and they're starting to see a little bit of 1,4-dioxanes around there. But the concentrations that we saw were relatively dilute. A lot of the mass that was remaining was associated with sort of these lower K zones. At one of the areas, it didn't look like that was necessarily contributing to elevated levels. So there was some good news in terms of that, but it did fit largely with the sort of new conceptual site model that we were developing, where some of that residual mass that's left over, stuff that wasn't necessarily removed earlier, is associated with those lower K zones and sticking around potentially a little bit longer.
Great, thank you. Is there evidence for 1,4-dioxane attenuation in anoxic zones as well as aerobic ones, given the common plume co-occurrence with CVOCs?
There is some evidence for attenuation. I'm not going to go and say that it's related to actual biodegradation. It may be related to other factors that are non-destructive. And so I don't think that there's anybody out there yet that's really has a lot of confidence that 1,4-dioxane would be degraded biologically, anaerobically. So I hope that I'm not making that implication that that's the case with these data. And then in terms of what we saw, we definitely saw a positive effect from the presence of dissolved oxygen at these sites being a positive indicator for 1,4-dioxane attenuation. So it's clear that those type of conditions favor attenuation.
Thank you. Did your data determine if historic remediation efforts impacted at all 1,4-dioxane attenuation and plume lengths in the field?
We could not establish that with any sort of statistical significance. That's not to say that it couldn't necessarily be done on a site-by-site basis, but on sort of the bulk data, it was pretty hard to do that with most remediation instances, with the exception of pump-and-treat systems. So the presence of a pump-and-treat system was associated in these particular cases with positive attenuation. So that was maybe the exception to the rule here.
Thank you. You reported that 1,4-dioxane source attenuation studies have led to you extrapolating some rates. Did you see evidence of 1,4-dioxane attenuation within plumes, or did you evaluate that at all?
That's a good question. We did to a limited extent. Most of the data that I presented was, as you said, was sort of source attenuation rates, which is a slightly different thing. But we did see evidence. So if you're moving along sort of the plume in the down gradient direction of groundwater, evidence for attenuation at some sites where we looked at it. That's actually a component of another project, a project that we're currently involved with Tony Danko at the Navy, John Wilson at Scissortail, Dave Friedman at Clemson, and Carmen LeBron, where we are going to gather some additional data on taking a look at those sort of plume attenuation rates, and that's sort of one of the critical rates that you need to understand, because it's what's controlling, whether you're going to see a plume attenuate over distance and be protective and sort of support selection of mNA. So good question.
Okay, great. And we have a specific question to one of the slides that you presented, which is slide 49, and the question is as follows. For the sites that you discuss here that were impacted with TCE, do you know if TCA was present or used?
I don't have that data in front of me, but I can, from what I recall, at most of the TCA sites, there was TCE present. The same wasn't necessarily true for the TCE sites, although there was a definite large number of them that also had TCA or one of the TCA byproducts present at them. So it's very clear based on what we know about, how these solvents were used, that TCE and TCA were used at a lot of these same sites at different periods of time, and the co-occurrence data, to my recollection, back that up.
Thank you. In your conclusions, you mentioned that attenuation is actually correlated with co-occurring contaminants. Can you please elaborate?
Yeah, I didn't present a lot of that data, but what we would see is that an attenuation, if you sort of plot the 1,4-dioxane attenuation rate versus, for example, the 1,1 DCE attenuation rates, where they were co-occurring at the same site, you saw a pretty linear type of a pattern develop. And so some of that might seem sort of counterintuitive, but what it's basically saying on our interpretation is that you sort of need to have the ability to remove 1,1 DCA at that site before you're going to see 1,4-dioxane be removed. So you can imagine at sites where you see a lot of 1,1 DCE attenuation, that basically clears the path for 1,4-dioxane to be attenuated, and then that may follow on from that. So we saw these sorts of relationships, sort of near linear relationships between 1,4-dioxane attenuation and attenuation of the co-occurring chlorinated solvents.
Great, thank you. And based on your conclusions about the viability of mNA at 1,4-dioxane sites and the importance of matrix diffusion, what additional site data, if any, would be valuable when you're trying to make site management decisions?
Well, we, as part of this project, were also able to collect data samples to send off for biomarker analysis at UCLA. We were able to send off samples for isotopic analysis that was done and coordinated with Peter Bennett at Haley and Aldrich, part of one of his sort of projects, and that work was done at University of Waterloo. And those data were really valuable for us in terms of being sort of additional lines of evidence because they showed that within sort of these low K zones, there was the presence of some of these biomarkers for 1,4-dioxane degradation. And there was evidence for fractionation of the 1,4-D. So those are the type of things that I think end up being important for sort of supporting any M&A arguments that are based again around sort of also demonstrating plume stability and lack of current risk to any receptors. So.
Thank you. And then one last question before we pull Pedro in. So a lot of the work that you talked about is for, or the data that you mined is for sites in California, where low threat closure is an approach at some sites. Do you believe that some of your data would support consideration of low threat closure approaches at sites with 1,4-dioxane, or should we limit those approaches for sites contaminated with hydrocarbons?
Well, I don't want to be an advocate for a particular strategy, but I do think that 1,4-dioxane is particularly applicable to situations where you have that kind of a low threat closure program like California does have for some of its contaminants. So I think that it's something that's definitely worth looking at, and it's definitely something that if you can build a strong enough case, that it's definitely worth considering and having a discussion about. So yeah, I think there is some promise in that.
Great, thank you. All right, so at this point, I'd like to broaden the discussion a little bit, and both you and Pedro talked about a very interesting research. But starting with you, Dave, what do you believe the next steps should be for research on this topic? And what are some follow-on research questions that have important implications for dealing with 1,4-dioxane impacted DoD sites? Dave?
Well, I think that a good first step, and I think there's a lot of people actually trying to do this, is to again go out and get the field data that's associated with demonstrating lines of evidence for M&A at these particular sites. Go out and determine what are the levels of particular biomarkers for 1,4-dioxane that you see and start compiling that data. And then use that data to basically, try to develop some sort of an association with whatever attenuation rates or patterns in concentration that you might be seeing at that particular site. And so that takes quite a bit of work to sort of do that and in terms of getting that data. But there's a number of different folks that are sort of working on that, and I think that there's potentially a lot of value in those efforts.
And Pedro, would you like to add anything to address this question?
Yeah, I think that I agree fully. I think that we also need to understand the diversity of dioxane degraders and their idiosyncrasies. Different bacteria have different preferences for different situations. It is really important not only to know what genes are there, but also who is carrying them so that they can express them and use those enzymes appropriately. And we need to understand better how these biodegradation capabilities are evolving and transferring from one species to their cousin and maybe to their distant cousin and so on, so that we can determine whether really biodegradation capabilities are being enhanced and increased with time. I think that's very important, but also we need to consider compatibility with other active measures that we might be taking, like in situ chemical oxidation and so on. And I think that there is a lot of opportunities also for material science for advanced materials to enhance the selectivity of many of these physicochemical treatment processes, such as in situ chemical oxidation, where we waste a lot of the oxidation capacity on things that react with water or with our material rather than with the targets.
Great, thank you. So in terms of take-home messages for our listeners, can you please summarize what lines of evidence are needed to demonstrate that M&A is a good choice at DoD sites, starting with you, Pedro?
Yeah, I think that it is very important to demonstrate that there is potential for biodegradation. For example, that specific degraders are present. Of course, what goes a long way is documentation of contaminant loss at the field scale, which sometimes you may have it. I mean, if you can show that your plume is stable or shrinking over years, if you have enough monitoring data, then that's great. Of course, that's not always possible, so you may need to rely on some geochemical indicators, including stable isotope analysis, seeing, for example, preferential depletion of 12C dioxane and enrichment of 13C dioxane left. Of course, microcosms also go a long way. We, for example, in our lab have been able to process samples from contaminated sites and not only do the biomarker analysis, but also a quick microcosm study and see degradation or even mineralization by using 14C labeled dioxane, which we have, that can help a lot. So these laboratory studies can also help, but overall we often need converging lines of evidence to be absolutely sure that natural attenuation is the right choice. And I think it's worth getting it, because if it is the right choice, you can save a lot of money in the end.
Thank you, Pedro. Dave, would you like to add anything?
Yeah, well, I mean, Pedro covered, I think, a lot of what's important. Maybe just add a couple things in. One is that, if you're going to go ahead and look for biomarkers for 1,4-dioxane degradation, definite recommendation to go ahead and look at biomarkers for CVOC degradation as well, because that's part of the story of what you need to sort of demonstrate here. And there's commercial labs available that'll do both. So, if you're spending the time to go out there and collect the samples, it seems worthwhile to go ahead and run a full analysis. And the second, just to sort of echo a little bit of what Pedro said, is that obviously for M&A you need those primary lines of evidence as well. And I've still talked to a lot of the people that sort of have some nervousness about even fully getting the 1,4-dioxane concentration data at a site and sort of trying to work off relatively limited data sets in order to sort of make a case. It just seems a little challenging. So it's to go ahead and make sure that you have the primary lines of evidence as well.
Great. And just to wrap up, one last question, and I'll have you start, Dave. We heard from both of you about unidentified sites that are likely impacted by 1,4-dioxane and likely for decades. Are you aware of any efforts by the Department of Defense or by regulatory agencies, including the states, to address this? And also, when would you recommend analyzing for 1,4-dioxane at DoD installations? Dave?
Well, my experience with DoD and talking with a lot of the folks is that they've been really proactive about doing this. And so I'm not sure I'd have any specific advice for those folks, because I think they're sort of already on board and sort of moving forward in terms of figuring out what the issues are and then what the management strategies are. But obviously, if there's sites where they don't fully have everything characterized, my recommendation is to go ahead and take the necessary efforts. Sort of at the state and federal level, I think there's still definitely a lot of work to be done, and sort of definitely probably a need to push it in certain directions. Pedro showed the drinking water data, and this is sort of a nationwide potential issue in terms of 1,4-dioxane. So I think it's something that we need to be more cognizant of.
Great. Pedro, would you like to add anything?
Well, I would like to say that I fully agree with Dave in the sense that everybody with the DoD that I have dealt with have been very proactive and very professional. They're very competent. So I think they know what they're doing. Of course, the more sometimes knowledge is power in a way. So the more you know about the sites that you're managing, the better that you're going to be. But we often need to not overreact and keep things in perspective. I mean, on a public health basis, sugar could be more dangerous than dioxane. But we do have a responsibility to, obey the law and meet remediation requirements and be proactive and, of course, prioritize our efforts on those things that pose the highest risks. Dioxane is certainly something important, and the more we know about this distribution and concentrations at our site, the better that we're at. So if we can do that as part of other monitoring efforts, I would certainly recommend proceeding accordingly.
Wonderful. Well, thank you both for two very good presentations. I would like to remind everyone that our next webinar is on Thursday, July 12th, and is titled Innovative Low-Cost Building Automation Sensors and Controls. This webinar will feature two speakers, Mr. Julian Lamp of Paragon Robotics and Dr. Michael Bramley from the Pacific Northwest National Laboratory. Registration is open for this webinar and other webinars, so please visit the CertaPen ESCCP webinar webpage for registration information. Before we conclude, I would like to remind you that both the audio and a copy of the presentation of today's session will be archived on our webinar webpage in case you would like to refer to them in the future. We would appreciate it at this time if you can please take a brief moment to complete the survey that will pop up on your screen. This concludes today's webcast. Thank you so much for your time.